Hello everyone, this is the Oasis guy with you, and today's topic is CRISPR-Cas9 genome editing tool. So today we're going to understand what is this process of gene editing, what is this tool used for, and also understand what components make this whole system. Okay, so we will start with the name. So what does CRISPR actually stands for? So it stands for clustered regularly intraspaced short palindromic repeats. So the name is quite long and maybe it's hard to understand, therefore we need to break it down into three main parts. So we would have the short palindromic repeats, they are regularly intraspaced, and they are in clusters. What does that mean? And in fact, understanding the meaning of these terms will allow us to understand the whole CRISPR system. And also, I don't want you to be surprised when I tell you that all these terms are actually related to bacteria. Why? So here we can explain it. So first of all, CRISPR is actually a natural defense mechanism used in bacteria, and which allows the bacteria to fight and break down any genetic material that enters the bacteria from any viruses. So here's the process. So what happens is that we have a virus that lands on bacteriophage, that lands on the bacterial surface, and it will try to infect the bacteria through injecting its genetic material into the bacteria. So if the bacteria manage to fight this infection and kill the virus, what happens next is that parts of this genetic material will be integrated into the DNA of the bacteria. These DNA will be then expressed in the future, and this will allow the bacteria to fight any infection from viruses that have similar DNA sequences, so the virus will be able to identify them. So let's zoom into the genome of the bacteria. So we can see here the bacteria is will have this type of genome, which has areas called spacers and areas called CRISPR repeats. So let's define both of them. So spacers are actually the genetic material of the past invaders, like viruses. So this area will be integrated into the bacterial genome, and they are called spacers. And then we would have the CRISPR repeats. So CRISPR repeats are actually short palindromic DNA sequences. And if you are asking what does palindromic mean, and also asking what the whole thing here means, so spacers with CRISPR repeats, they are both called CRISPR locus. So this whole area of the DNA, it's called CRISPR locus. So what we have here is that we would have the word palindromic. It means that the sequence will be read the same from both sides. So read it from 5' to 3', it's the same sequence read 5' to 3' in the antisense. And these are the CRISPR repeats. Okay, so after seeing how the genome of the bacteria looks like, so now we can go to start showing what does the name of CRISPR means. So we would have clustered regularly intraspaced short palindromic repeats. So the short palindromic repeats are here referring to the CRISPR repeats we have just shown in the bacterial genome. So these are palindromic repeats, and these repeats are actually spaced by something called a spacer. So spacer is the DNA of the virus, as we saw in the bacterial genome. So short palindromic repeats, it refers to these repeats, and regularly intraspaced, which mean they are spaced by the viral DNA. This pattern, we will see it in clusters, and this is where cluster comes from. So that's mainly what the name of CRISPR means, and hopefully this makes it much clearer. And the whole thing here, it's called CRISPR array. So you might see it in a textbook or in another video. If you see a CRISPR array, this is what it refers to. It refers to the repeats, palindromic repeats with the spacers, which are the viral DNA. So in a bacteria, when a virus invades a bacteria, like a bacteriophage, so what happens is that these CRISPR arrays will be transcribed into an RNA. So this RNA is called a guide RNA. So gRNA, which is a guide RNA, and these RNAs will have a sequence here, which corresponds to the spacers, which are the viral past infections, the viral sequences of past infections. So we will go into more details into the structure of the guide RNA in the next slide, but here we will just show the general process. So what happens is that once the guide RNAs are transcribed, they will bind to proteins called Cas proteins, which are endonucleases that are able to cut the DNA. So once the guide RNA bind to the Cas9, both of them will start scanning the DNA. For example, in this case is the viral DNA, okay? And once the sequence in the guide RNA identifies the target sequence, the guide will bind to it, and then the Cas9 will cut the DNA. And what happens then is that the DNA will be fragmented and cleaved and inactive. And this will make the whole virus DNA ineffective, and the bacteria will not die. Okay, let's dive deeper now into the structure of the guide RNA, as it's a very important part in the CRISPR-Cas9 system. So first of all, as we said before, that the CRISPR array within the bacterial genome will be transcribed into a guide RNA. The guide RNA at first, it will be something linear like that. However, this linear structure, it's very unstable, so it's will induces the guide RNA degradation. Therefore, the RNA will be folded, will fold in itself to form this duplex structure. Okay, so this duplex structure plays two main roles. So the first role is that it stabilizes the whole guide RNA, and also it allows it to bind to Cas protein. So in this form, guide RNA cannot bind Cas proteins. However, in this form, they are more stable and they can bind the Cas protein. So this structure is separated in two main parts. Okay, the first part, which is this one on the right, and it's called the transactivating CRISPR RNA or tracer RNA, and this one it's called the CRISPR RNA. Okay, so we have the CRISPR RNA and the tracer RNA. CRISPR RNA, they are characterized with carrying the spacer sequence, which is the one that is corresponding to the DNA sequence of the virus, and also it, as we discussed before, that it targets the Cas9 to the target sequence. Okay, and this transactivating RNA, it's very important because it stabilizes the whole structure, and also without it, the guide RNA will not be able to bind to Cas protein. So the tracer RNA are responsible of facilitating guide RNA binding to Cas protein. Okay, so after now understanding the structure of the guide RNA and how it's guided to the right position within the DNA, now we need to understand something more about the Cas protein and something called the PAM sequence. So as we know here that we already know now that the guide RNA will bind the Cas protein, and then these two structures, or this complex, will start scanning the DNA, and in this case is the DNA of the virus. They will start scanning the DNA looking for a sequence that is complementary to the sequence in the guide RNA. Okay, as they keep scanning, once they find the target sequence, the guide RNA will bind to it, the DNA will be unfolded in that area, and the Cas protein will cut. However, before the Cas protein will be able to cut the DNA, they need to identify a small DNA sequence called the PAM sequence. So the PAM sequence is a very short DNA sequence, very close to the target DNA, and in this case is the DNA of the virus. So it's going to be very close to the target DNA virus, okay, to that target sequence. So once the Cas protein identifies the PAM sequence, then the Cas protein will start the cutting of the DNA. However, we need to understand that without the guide RNA being able to identify the target sequence, or without the PAM sequence, the Cas protein will not cut the DNA. So these two things are very important in this sense. Okay, just always bear it in mind. The Cas protein needs to identify the PAM sequence, and the guide RNA needs to identify the target sequence in the DNA as well. Okay? So now, after understanding the CRISPR-Cas system in the context of bacteria and how it's used as a defense mechanism against bacteriophages, now we want to study CRISPR-Cas9 as a gene editing tool. So here we're going to talk mainly about a Cas9, which is a specific type of the Cas proteins, and we will see how these genetic tools are used in the research field. Okay, so CRISPR-Cas9 as a gene editing technology, it's most widely used to knock out gene expression. And when I say knock out, it means that we prevent a specific gene from being expressed. So for example, if you have a protein of interest that you don't want a specific cell line to express that protein anymore, so what you do is that you use CRISPR-Cas9 to knock out that gene expression. So I will show you now in steps how this process occurs in a very general way. So first of all, as we said, you need to identify a gene of interest that you want to knock out. Once you identify that gene, you need to determine the DNA sequence of that gene, specifically the exon sequences of that gene. Once you do that, you will be able to design a guide RNA that has a specific sequence that is complementary to the DNA sequence of your targeted gene. Okay, so once you'll be able to do that, the guide RNA, it will be guided to that sequence in specific. Okay, so once you design the guide RNA, usually in the research field, you send the sequence of your gene of interest to a specific biotechnology company, and they will just make the guide RNA for you and then send it to you. Then once you get your guide RNAs, you will clone them into a vector. Okay, so these vectors, they are usually having already or they need to have already a Cas9 protein expressing gene in them. So once this vector is introduced into your cell of interest, what happens is that this cell will start producing Cas9 and the guide RNA. Okay, and then the whole process starts again. So the guide RNA will bind to the CRISPR-Cas9 to the Cas9 protein, and then they will be targeted to that specific sequence. So once you knock out the expression of your gene, of course, some cells, they're going to be knocked out and some cells were not going to be knocked out. So so you need to find a way and do some screening to distinguish these cells that have the knockout from the rest of the cells. So once you do the screening process, for example, usually these vectors would have an antibiotic resistant. So for example, these cells that take up the vector will be resistant to a specific antibiotic, and therefore all the other cells will die but these cells. After doing that, you need then to confirm that the cell that you selected has a knockout gene of interest. So you do that by either PCR, which will allow you to identify any insertions or deletions that are introduced in your gene of interest, or you can use qPCR, which is to determine the knockout at an mRNA level, or a Western blot, which is the most commonly used one in this case, and that will confirm that the protein is no longer expressed. All the process from 2 to 6 and all the techniques used to confirm the knockout of the gene, they are all whole experiments on its own, every one of them. Therefore, I will be doing future videos on every single study and every single experiment, but here I'm just showing them very generally to understand the whole process of how to knock out a gene. Also, you might be asking yourself why the CRISPR-Cas9 system results in a gene knockout. So to answer that question, we need to go through the cellular mechanism that is repairing the double-strand break that occurs in the gene. So as we know that once the guide RNA and the Cas binds to the target sequence, then the BAM sequence also is identified, then the DNA will be cut, and then we would have the double-strand break. So the DNA doesn't stay like that. So what happens is that the cell will try to repair this mutation or this double-strand break, which would lead to a mutation at the end. So the process of doing that, it's called non-homologous enjoining. So it's a very common cellular mechanism, mainly used to repair the double-strand break. And usually, once the two strands are joined together again, what happens is that some kind of insertions of new nucleotides or the deletion of some nucleotides will occur, and therefore the new sequence here will be different to the original before the double strand break, and this would result in knocking out the expression of the whole gene. It is also important to highlight that CRISPR-Cas9 gene editing technology has been used so extensively in the research field, and therefore it has been also used a lot in preclinical studies because it has a lot of potential therapeutic targets and applications. In fact, just a few months ago, the UK approved for the first time a CRISPR treatment for diseases, for two main diseases, which are sickle cell disease and beta thalassemia. And I will make future videos on how CRISPR was used in treating these diseases. But this is very promising and showing that CRISPR maybe in the near future can also be used in some cancer therapy, hopefully. And here is some questions to test your understanding of this topic. Okay guys, we came to the end of this tutorial on CRISPR-Cas9 genome editing technology. I hope you enjoyed the video and it was beneficial. Please don't forget to like, comment and subscribe. And also I would recommend that you activate the notification, so hopefully when I upload my future videos, you'll be able to get notified. Thank you.